This will be a short video describing probability in science, and particularly the biology lab when we study genetics. These are from my high school biology days. This was actually a college class that I taught, and this was the lab I had when I was in the Azores, one of the islands pertaining to Portugal in the Atlantic Ocean. And these were military personnel who, in the evenings and then on the weekends, took a biology class. So as you can see here, we're dealing with chromosomes. And this is a double-stranded chromosome. And this is the centromere. And then we have the dyads here, the double strands, where we can have crossing over. And in genetics, there is a percentage of probability that a particular gene will cross from one position on the chromosome and switch to the other. And it would take all the other parts of it together. Now, that gets to be rather complicated. Ours is going to be simple work. But this was a project they were working on. And another shot, and more people. This was the daughter of this lady, and she just came along to see what was going on. And there's some more. These all had to do with genetics and crossing over. And by using the models, you get to see how it's done, and then relating to its probability of crossing over, we can see where traits are linked on the chromosomes. Now, another lab that we did, that we'll get to very shortly here, is this one in which we had fruit flies. Now, this box that you see here in the front is what we call genetic corn. Now, most of you, when you think of corn, you think of a yellow colored corn or a very light colored corn. Well, these are recessive traits, and corn is bred for these particular characteristics. Now, a dominant color in corn, believe it or not, is black. So we see we are crossing a purebred black with a purebred yellow here. Now, the offspring are all black, and each kernel on the corn cob is an offspring. So in our F1 generation, the phenotype, the appearance of the corn is black. Now, when we allow members of this F1 generation to cross, an interesting thing happens. Now remember, they're both black, but the offspring are mostly black, but now there are some yellow. This is the F2 generation. Now, we know mathematically what the ratio should be, and it would be 3 black to 1 yellow. But now, empirically, they are looking at this corn cob, and they will physically count them to see if, in fact, the data they get from counting a specific example, in this case, this corn cob, F2 generation, has 3 black for every 1 yellow. So that's what we do in biology lab. And if you're going to take a biology course, you'd probably do something like that. And that's where probability comes in. Now, in these little jars with the sponge caps here, we have fruit flies, Drosophila melanogaster. And again, we can cross purebred normal winged fruit flies with vestigial wing fruit flies. the wings, instead of being normal shape, they're all shriveled up, and these flies can't fly. Now, when we do that cross, we get an F1 generation where all the flies are normal. But when we allow the F1 generation to cross, the offspring, the F2 generation, again, theoretically, should show a 3-to-1 ratio, normal wings to vestigial wings, and then with a very delicate paintbrush, after you've etherized the flies, you can do a physical count. So we're comparing classic probability to empirical probability, and that's another experiment that we do. Okay, and there you see just another view of it. When I taught biology, I had lots of equipment and charts and models, and again, we had a pretty good course because all this was available to my high school students as well. Here she's looking at the flies. And of course, you have to be careful when you take the cap off, the flies will just fly around. So there's a funnel, and then there's ether, and often a few flies escape. And this was my fish tank, and we'd use the carcasses of the flies as fish food. This is one of my colleagues that would come in to work with me. And we're moving on. We did some electrophoresis as well. But I want to get to our last one. Here we were doing a protein synthesis. This is the last one I'm going to show you. Because now in this big bowl of little cylinders, half were red and half were white. And we'd kind of shake them up and mix them all up. And then the students would dip in without looking, just get their hand in that big bowl and pick out, feeling them, 2 cylinders. And then they would put that on the table as you see here. And what we were doing is we expected, since they were half red and half white, we would have expected a 50-50. But then we also were interested in the order that they were picked. And then, after 1 round, 2 rounds, usually after 1 round, we didn't get 50-50. But after 10 rounds, it got closer and closer to the expected probability. So again, this was empirical, but we were comparing it to what classical results would give. Okay, just a little quick something here. Didn't want it to be too long, but hopefully you have an idea of how probability is used in biology. All right, we'll wind this up.